Hi everyone. In this section, we will talk about antigen and antibody. After watching this video, you should be able to tell the differences between antigen and antibody. The term antigen refers to any self or non-self substance that elicits an immune response. Antigens include molecules such as proteins, nucleoproteins, polysaccharides, and some glycolipids. Most antigens are large, complex molecules with a molecular mass generally greater than 10,000 daltons. Each antigen can have several antigenic determinants called epitopes that stimulate production of and combine with specific antibodies. Let's take a look at the definition of antibody. It refers to a blood protein produced in response to and counteracting a specific antigen. Antibodies combine chemically with substances which the body recognizes as alien, such as bacteria, virus, and foreign substances in the blood. The strength of binding between the antibody and the antigen at a single binding site is known as the antibody's affinity for the antigen. In other words, antibodies are specialized proteins that travel through the bloodstream and are found in bodily fluids. They are utilized by the immune system to identify and defend against foreign intruders to the body. An antibody or immunoglobulin is a glycoprotein. All immunoglobulin molecules have a basic structure composed of 4 polypeptide chains, 2 identical heavy and 2 identical light chains. There are 5 classes of human antibodies. One of them is IgG, which can be further classified into IgG1, 2, 3, and 4. The other 4 classes are IgA, IgM, IgD, and IgE. All these antibodies share a basic structure that varies in order to accomplish specific biological functions. Distinguish between self and non-self is essential in maintaining host integrity. This distinction must be highly specific and selective so invading pathogens are eliminated but host tissue is not destroyed. The recognition of foreignness in mammals is accomplished by the major histocompatibility complex. MHC molecules are cell surface proteins coded by a group of genes termed the major histocompatibility complex. The class 1 MHC proteins are found on all nucleated cells of mammals. The class 2 MHC proteins are expressed on macrophage dendritic cells, and B cells. The human MHC genes products are called the human leukocyte antigen. Class II MHC molecules are produced only by certain white blood cells, such as activated macrophages, dendritic cells, mature B cells, some T cells, and certain cells of other tissues. Importantly, Class II molecules are required for T cell communication with macrophage, dendritic cells, and B cells. Class II MHC molecules are also transmembrane proteins consisting of two distinct chains. Similar to MHC class I, the protein chains expressed from MHC class II genes combine to form a three-dimensional antigen-binding pocket into which a non-self peptide fragment can be captured for presentation to other cells of the immune system. Although MHC class I and class II molecules are structurally distinct, both fold into similar shapes. Each MHC molecule has a deep groove into which a short peptide derived from a foreign substance combined. Antigen fragments in the MHC groove must be present to activate T cells, which in turn activate other immunocytes. Exposure to an antigen by an individual for the first time is called primary immune response, and re-exposure to the same antigen is called secondary immune response, which develops due to immunologic memory. After the first exposure, antigen-specific memory cells are formed, which are responsible for immunologic memory. The primary antibody response in a host occurs following initial exposure to the antigen. This response has lag, log, plateau, and decline phases. Upon secondary antigen challenge, the B cells mount a heightened and accelerated response. Finally, let's take a look at the keywords in this section. Okay, that's all for this section.